This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 118, recorded on December 28th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there.
Hi, Elio. How are you?
Well, I got probably a positive strand RNA virus. In other words, I got a common cold.
You know the polarity of the genome.
If I sound funny, it's because I feel funny.
Yeah, you do sound a bit different, but that's what these viruses do to you, and you will spread it to someone else for sure.
In a minute, let me say something about it after you introduce.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Dedicated to podcasting, we are the last week of 2015.
That's right. So we can wish our listeners a happy 2016, can't we?
That's right. The next time we will record will be 2016, an even year. I like even years. And I will be a year older because my birthday is January 2nd.
All right.
And but maybe not wiser. But we podcast all the time, and last week I did a TWiV, and this week I did a TWiV, and people say, why don't you take a break? But I don't want to. There's too much to talk about. Elio, tell us how you got your cold.
Well, grandchild came from Maryland. I live in San Diego, and in the process, he brought the virus with him, probably, because he came with a cold. But let me say something about a thought I once had about the common cold. You know how, well, when I have a cold, my wife thinks I've become a bear. I mean, all I want is hibernate. I don't want to deal with anybody, even not with her. And so I was wondering, what is that? I think other people report the same thing. When they get a cold, they don't want to be around people and so forth. So I'm wondering if that's not a biological mechanism designed to avoid transfer of the virus to other people. What do you think?
So a host defense.
A host defense, right.
Not viral encoded.
Modified, but behavior is modified by the infection.
Yeah. It would certainly not be a virus thing, Michele, right?
Right.
And it's fairly specific for the cold. If you're sick with other things, you don't quite feel the same way. I think it's all these cytokines that make you feel lousy. That tell you, don't talk to other people.
Go lay down somewhere.
You know, it sounds plausible, but it's nothing we could ever test, right?
Oh, you want tests too?
I do. I want to validate.
I think you could test it.
How
You're
going to spoil a good idea by doing experiments.
Yeah, take an animal and inject a certain cytokine in the absence of the virus and see if that cytokine makes the animal want to go lay in the corner.
Right. Yeah, that can be done.
I suppose animals, in my view, they want to lay in the corner all the time anyway. But yeah, I suppose you could find the appropriate animal. Michele, what is Ann Arbor like during these holidays? Is it quiet?
Yes,
curious.
it is quiet.
All the students are gone, right?
That's right.
Is it festive?
Well, no, it's very still, and I think everybody's gathering with friends and family in their homes.
Yeah.
So, yeah, it's been great. And today we got a wintry mix.
Snow?
Snow, and it's going to turn to sleet. And so I walked into work today instead of taking my bicycle.
How long does it take to walk? Half hour?
Oh, no. Yeah, not even.
Oh, that's great.
It's just about a mile.
Well, we've had crazy warm weather until today, actually.
Yeah.
It's been like in the
60s. It was warmer in New York City than in San Diego for a while.
Yeah. See, we're the new San Diego, Elio. Everyone's going to come here now.
Yeah. It was 60 degrees here, too, last week. And I honestly think that Michiganders are more comfortable with the wintry mix. The warm weather was making us all a little bit uncomfortable, uneasy.
Michael Schmidt was supposed to join us today, but he had a 3 p.m. flight, which would have worked, which they moved up on him. So he's flying right now, unfortunately. But, you know, traveling these days is tough. It's the weather. You're at the mercy of the weather, right?
And there's not very many extra seats on the planes.
Nope.
No extra capacity.
All right. Today we have two stories for you to round up 2015 on This Week in Microbiology. We're going to start with a snippet by Elio, which is really cool. It's all about pack hunting, right, Elio?
It is. It's pack hunting by amoebas. And what do they hunt? But nematodes. And I gotta tell you, I've been worried about the fact that nematodes are terribly important and terribly common, and they exist all over the place, and they're fascinating, and we don't highlight them very much in microbiology because they are, of course, multicellular organisms, and we don't deal with multicellular organisms all that much. But it's a mistake because they have a lot to do with the microbial world. So let me say something about nematodes in general. May I?
Yes. I just had a thought when you were saying this. That's why we should call it microbial sciences. Then we could include the nematodes, right?
Absolutely, absolutely. Well, I'll tell you, they have, first of all, they are pathogens, hookworms and nematodes, stuff like that. Mm-hmm. In addition, they're extraordinarily common. There are about between 1 and 10 million nematodes in a square meter of soil. They're the most abundant animal in oceanic sediments, and they've been found in gold mines 3 to 5 kilometers down, in the water. It's amazing. So that they are really amazing things. And they're tiny, of course. They're about, 1 millimeter across, something long. In addition, they are great for research because one of the fantastic model systems is C. elegans, which is a nematode. They are also, and C. elegans itself is a great model system for pathogens because it can be infected by a whole lot of bugs. Pseudomonas, I think, is one of the main ones that's used. And on top of that, nematodes eat bacteria. Now, the subject of eating bacteria, that's another subject that I'm hung up on. It seems to me we know so little about. Phages eat bacteria, nematodes eat bacteria, amoebas eat bacteria, all kinds of things eat bacteria. And we don't think of bacteria as food normally.
Unless you study Legionella, like I do.
Right.
Amoeba are the natural reservoir for that human pathogen.
Okay. Well, I'll tell you, the importance of nematodes came to me, and I read a quote from somebody, N.A. Cobb, 1914, and I like to read it. If all the matter in the universe, except the nematodes, were swept away, our world would still be dimly recognizable. And if, as disembodied spirits, we could then investigate it, we would find its mountains, hills, vales, rivers, lakes, and oceans represented by a thin film of nematodes. The location of towns would be decipherable, since for every massing of human beings, there would be a corresponding massing of certain nematodes. Trees would still stand in ghostly rows, representing our streets and highways. The location of the various plants and animals would still be decipherable, and had we sufficient knowledge, in many cases, even the species could be determined by examination of their erstwhile nematode parasites. So, if everything else went away, the nematodes would take over.
I thought the cockroaches would take over.
Well, it depends on how much radiation you use to affect this.
Right.
Anyhow, have I sold you on the importance of nematodes?
For sure, yeah.
All right. So, now, I say they're common in soils. They're among, in their size range, they're certainly the most common multicellular entity. Bigger ones like earthworms make up probably more of a biomass, but nematodes are very, very important in the soil. And the soil is one phenomenal place in complexity. Boy, we think about human pathogenesis, interaction with bugs as being complicated. It gets at least as complicated in the soil because there's so many players, such a heterogeneity in the environment. Sometimes it's wet, sometimes it's dry. It always consists of solids with some water interspersed. It's really very, very complicated. And I would say that the whole subject of soil microbiology, which has gotten a lot of attention, of course, through the years, in fact, early days, microbiologists were soil microbiologists like Vinogradsky. But still, I think there's much more to learn. So here is an interesting fact that was uncovered in a paper by Geisen, Rosengarten, Koller, Mulder, Ulrich, and Bonkowski from Germany, the Netherlands, and Austria, various institutions. These are I can tell from the department they call the Department of Terrestrial Ecology, and stuff like that. Anyhow, it's a nice little paper which appeared in Environmental Microbiology, and what it says is that nematodes are not only predators, for example, to bacteria, but they are prey. And they are prey from a fairly common amoeba in the soil called Cryptodiflugia operculata. I don't know what crypto means, but I know what it means, but I don't know what it denotes in this connection. But Diflugia are testate amoebas. Now, testate doesn't mean, I don't know what you think it means, but it means that they have a covering. They are shelled amoebas. They have a shell on top of them. And someday we ought to talk about how this has been used in the early days to uncover a phenomenon of epigenetics. But we won't talk about that now. Anyhow, Cryptodiflugia, which is a fairly common soil organism, has a specialized way of finding nematodes and eating them. Namely, it hunts as a pack. And there are phenomenal movies in this paper, which I hope the readers go to it, and we have those in our show notes. And what they show is that this nematode gets attacked at the end. And obviously, a nematode has two ends, right? It gets attacked at both ends by one or a few amoebas, and then more come, and more come, and more come. And before you know it, bingo, it's gone. The nematode is all eaten up. So this is predation by pack hunting. And it's not known to what extent this affects the stability of the nematode population. And nothing is known yet about the signals involved. It looks like the amoebas recognize the nematodes at a distance. They further recognize which are the ends of the nematode. All these are possibly due to signals, chemical signals. It's complicated because there are some products of amoebas, different amoebas, that repel the nematodes. So they do the opposite of what they do here. Here they want to attract the nematodes, or at least be attracted to the nematodes. Amoebas are not the only ones that do this. Flagellates also kill nematodes. And so here we have both things are bacteriovores. The nematodes eat bacteria. The amoebas eat bacteria. In this case, one eats the other, and the bacteria are sitting there laughing and laughing. They're happy.
Licking their chops.
Licking their chops because they're not being eaten. Anyhow, the population of nematodes varies with moisture, but amoebas can move even under reduced moisture conditions. So they are likely to find the nematodes under a whole variety of conditions. So that's pretty much the paper. They determined by using 18 sRNA sequences the frequency of these amoebas in various places. It's most abundant in forest soils and forest litters. Then comes grassland soil, and then peatland soil. You'd think that's a good place for living things to wander around, but these particular amoebas are not that common there.
Elio, if they just added one amoeba to a nematode, would that single amoeba be able to attack and eat the nematode?
I don't know the answer. I don't know. The paper doesn't show that. The paper only deals with a horde,
right? With
a pack.
Yeah, they had a bunch of the amoeba, but I'm wondering.
Yeah, that's what they show you. Surely there must be cases where a single amoeba finds a single nematodes, and what happens then, I don't know.
So is it a case of helping each other, or one finds it and the others say, hey, look what he got or she got. Let's go get some of the same thing,
Well, it sounds like from studying the movies, they found that a key step was one amoeba would latch on to the tail of these worms that can squirm through the soil. And in doing so, would inhibit its movement so that nematode couldn't just slither away. And then that gave the other amoeba a chance to latch on.
So it becomes it's
a group effort.
Yeah.
So I guess that one amoeba. Yeah. So, I'm interested to know if the one amoeba could eat the nematode, or if the others help to do it, or if they all just take this is kind of a symbiosis, right? They're helping each other. Well, pack destruction.
Pack behavior. Yeah.
Behavior.
Yeah.
I wonder, too, if the amoeba might have a toxin, like a neurotoxin, to stop the movement of these nematodes.
Does. It must have something like that, or else the nematode stops moving on its own once it's infected. But once it's affected, but it's very likely that this is the kind of paralyzing that scorpions do when they prey. You know, you don't want the prey to move away if you're a predator.
So then the amoeba must somehow signal the other amoeba, right? Chemically, I suppose, to get them to come, right?
Well, I would bet that there's a sequence of signals.
Yeah.
Some recognizing
maybe a pheromone
the nematode.
released by the worm.
Yeah, that could be also the worm might be releasing something, yeah.
The what? I'm sorry?
Maybe the worm releases some stress pheromone that attracts the amoeba.
Mm-hmm.
Right.
But just to give the listeners an appreciation of the scale, each nematode, I think it would take about, 20 or 30 of these amoeba to decorate its whole surface. So there's quite a difference in size.
Yeah, I'll say.
And the amoeba are phagocytic, right? They are basically taking up pieces of this worm.
It's not easy to be a nematode. Not only get you eaten up by amoeba, you get eaten up by fungi. You know about these fungi that make lasso-like structures of 3 cells in a circle, and they attract a nematode. And when they do, the cells inflate, becoming like tightening a noose.
Wow.
And the nematode still tries to wiggle.
Wow.
It cannot wiggle away. And then the fungus sends in a specialized cell called a haustorium, which essentially sucks the nematode dry.
I like that title. It's not easy to be a nematode. You could write a little book one day, Elio, about nematodes and call it that.
All right. Okay.
Just a short book that you could read in a few hours and would be very interesting with all kinds of stories like that.
How about that?
People would probably buy it. It would
a new project for 2016.
And now that you're finished with your microbe book.
Right. I don't have enough to do.
Yes.
Michele and I, for the readers, have been involved in editing a new edition of a textbook in microbiology. We've been rather busy, haven't we?
Yeah, not just editing, but a lot of new content.
That's right. There's a lot of, that's fair.
But it's been worth it because not only are you going to educate people, but you're going to become...
Not often do you get the pleasure of working with somebody as wonderful as Michele and Hema.
And our other colleagues.
Hema.
Yeah.
She's wonderful. So it's been a lovely collaboration.
I've learned a lot.
Are you looking forward to becoming wealthy?
Are you kidding?
I know, I'm joking because I know I wrote a book.
If you write Samuelson Principles of Economics, you can get rich.
That's right.
Anything else in textbooks?
You have to write a book that all the freshmen in the U.S. use.
That's right. That's right.
So economics.
This is not it.
No.
We missed it, Michele.
I'm telling you, do the nematode book. Some nice illustrations. Well, thank you, Elio.
Sure.
I like that story. That's really cool. This episode of TWiM is brought to you by ASM Microbe 2016. Hurry up. You have just until January 12th to submit your abstract for the all-new ASM Microbe 2016. This will be taking place from June 16th to 20th, 2016, of course, in Boston, Massachusetts. It'll feature the best microbial sciences in the world and will explore the full scope of microbiology. It'll be a distinct forum for cross-disciplinary study, both in the lecture hall and the integrated exhibit and poster hall. It's a unique meeting designed to spark collaboration and discussion. If your work touches the microbial sciences, this is the meeting you should attend. Learn, connect, and advance at the first ever ASM Microbe 2016. This is a merging of the previous ASM General Meeting and ICAC all together under one roof. Lots and lots of people who are interested in microbiology. And I will be there. I will be doing both a TWiM and a TWiV. In fact, the Friday of the meeting at 2:45 p.m., we will be recording TWiM, and the following Saturday at 8:15 in the morning, we'll be recording TWiV. Wow, that's pretty early, but we'll be there. Come check it out. It'll be on the program. If you want to send in your abstract, again, the deadline is January 12th. Or if you just want to register for the meeting, you can go to asm.org/microbe2016. That's all one word, asm.org/microbe2016. We'll see you there. All right, we have a paper from PLOS One, which I discovered not too long ago, and it is called Mushrooms as Rainmakers, How Spores Act as Nuclei for Raindrops. And the authors are Hassett, Fisher, and Money from Miami University in Ohio, not in Miami, Florida. This is
not that Miami University.
This is the one, it's a little cooler here in Oxford, Ohio. And so we are returning to mushrooms. We're not going to pick them and we're not going to kick them today, but we're going to be amazed at what they do in the ecosystem. And first, I want to talk a little bit about mushrooms and mushroom spores. Probably everyone knows that mushrooms are the fruiting body of a fungus. So there is a part honor in the ground called the mycelium, which is made up of many, many hyphae all joined together. And these can be quite large, as we've talked about before. They can be acres and acres in size. And the mycelia send up mushrooms to the surface in order to reproduce, to make spores. So the mushroom, its function is to make spores that will then go on and allow the growth of more fungi. All right, so mushrooms make spores. That cap, underneath that cap are, and some mushrooms, not all of them, are a number of what we call gills. And in the gills, those lines that you can see, if you go to the supermarket and buy a mushroom and turn it over, you'll see those gills. They contain structures called basidia, that's plural, or basidium, which literally means a pedestal, little pedestal. And they are the structures on which the spores develop and eventually fall off and fall to earth. And when the conditions are right, they germinate, they form new hyphae, and you have a new mycelium form. So this is the reproductive structure of the fungus. Now, an individual mushroom, one mushroom with its gills underneath, can release 30,000 basidiospores each second, every second.
Wow.
That's billions in one day. That's just one mushroom. Basically, there are so many spores floating around the air because of this, and we'll talk in a moment about how many exactly, but there are millions of tons of fungal spores released every year, and we can't even see them. These are microscopic entities, these spores. So they
are, compared to bacteria, they're more like the size of a red blood cell, say.
Right. 5
to 10 microns across, something like that.
But we can't see them, and the air is full of them. Wherever you are, you're breathing spores. Even in houses and buildings, there are fungi that produce spores. Not as much as in a forest, for example, but there are lots of them. So the air is full of them.
Those are different spores, by the way. The spores you're talking about are the result of sex, whereas the spores that the mold makes on an apple are called conidia. Those are asexual spores made vegetatively. So this is a difference. But both are reproductive structures. They're not as tough as bacterial spores, which can resist boiling, but they're tougher than their vegetative cells.
Now, these spores, basidiospores we call them, because they're released from a basidium, the little pedestal, they don't just fall out of the gill. They're actually discharged, and the authors call it by a catapult mechanism. So what powers the catapult? It's a movement of a drop of water over the surface of the spore. And this is really cool. So basically, the spores are shot out of the mushroom, out of the gill, by the movement of a drop of fluid. And this drop is called Buller's drop, and it's named after a scientist, A.H.R. Buller, who is called the Einstein of mycology. Did you know of Buller, Elio, since you're a mycologist?
Yeah, he was a British scientist who settled in Manitoba, Canada, and he wandered around with blinders because he doesn't want on the street. He didn't want his eyes to get losing the dark adaptation he needed, stuff like that. He was a character.
Sounds like a character. So this drop is named after him. So what happens is when a basidiospore is ready to be released, on the surface, in the cell wall, actually, of the spore, there are sugars which serve as condensation loci for water vapor in the air. So
hygroscopic.
They're hygroscopic, exactly.
And the image that I'm sure many listeners will be familiar with is if you've seen a stick of cotton candy that you get fresh, and then after you've walked around with it for a bit, you'll see little beads of glistening moisture.
That's right.
And it's the same principle. The sugars attract water from the air.
Yep.
Nice analogy.
Very good. The cotton candy is great, yeah. And of course, if you're in a dry place, it won't happen as quickly. So in order for these spores to come out, you have to have enough water vapor in the air, and that'll be experimentally important in a moment. Now, this is an open access article in PLOS One, so everyone can go see it. And I suggest you look at Figure 1, which is a lovely diagram of the formation of Buller's drop on the spore. This drop of water forms at what's called the hilar appendix. It's a little point at the bottom of the spore where it attaches to the basidium. So Buller's drop of water begins to accumulate there. And then just above this hilar appendix is a flat surface of the spore. And there another bit of water starts to accumulate. This is called the adaxial drop. And as that drop...
Say it slowly. Adaxial.
Adaxial. You don't want me to say adaxial?
Adaxial doesn't convey that.
Adaxial. The adaxial drop, and this is a flat area of water. It's not a spherical drop as is Buller's drop. And as these two bodies of water get larger, they form independently. The adaxial drop eventually gets too big, and it slides down the surface of the spore. It merges with Buller's drop, and then this changes. When these two bodies of water, they call them, although when I say body of water, I think of an ocean, but this is a body of water. The release of surface tension and the change in the center of mass launches the basidiospore out of the mushroom gill. And the speed is quite remarkable, 1.8 meters per second. So this water,
amazing, isn't it?
The water just slides down the spore, and for every action, there's an opposite reaction. The spore goes shooting off out of the gill. And, of course, it falls out of the gill, and then there's air flowing around the mushroom. It gets caught up in that. And then, of course, the fluid evaporates, and the spores can fly off quite long distances and eventually get into the soil.
Let me add a couple of things here.
Yeah.
First of all, the acceleration of the spore is something like 10,000 Gs. It's unbelievable.
Wow.
Absolutely unbelievable. NASA would dream of it, I think. Anyhow, it only lasts for a very small fraction of a second. But the other thing, with this force, you wonder if it's made on the surface of a gill, how does it keep from hitting the gill across? Right? And the answer is that the inertia of the particle is sufficient so that it doesn't travel very far. It starts out at a terrific speed, but it doesn't sustain it for very long. Yeah. And after a while, it just hits the viscosity of air and it falls down. Right. Now, this trajectory, which is, if you can picture it, going out of the surface of the gill, it goes out horizontally, then suddenly it drops. That's the ultimate slider. Any baseball pitcher would kill for that.
Yes.
Yeah. Anyhow, but this way, it clears the next gill. It falls underneath the mushroom and on the forest floor. This is not a highly appreciated fact. On the forest floor are breezes, and so they carry the spore aloft. That's the
Somehow
story.
I want to bring Mary Poppins into this because I'm picturing her with her umbrella and rain, and then suddenly she goes aloft, and that's what I'm thinking of with these spores.
Yep.
Yeah, that's right. Very good.
Right. Of course, losing the water is part of it too, right? They get lighter because the water is quite a big part of its mass, I would think. So these spores are a big part of the air, especially above tropical forests, where there's lots of rainfall and there's a lot of material in the soil for fungi to grow in. In fact, one of the components of the cell wall of fungi, mannitol, which is a 6-carbon sugar alcohol, can be used to measure the number of spores in the atmosphere. And one estimate is that there are 50 million tons every year of spores dispersed into the atmosphere, which corresponds to 1,000 spores for every millimeter of the Earth's surface. Just take a look at some area right next to you that's a millimeter square, and there'd be 1,000 spores per each one of those. That's how much 50 million tons is. Now, these are not the only particulate substances in the atmosphere. You have plant spores, you have pollen grains, so you have lots and lots of particulate biological particles, let's call them. And these are thought to serve as nuclei for cloud formation and rain, especially in places where there's a lot of forest like the Amazon basin. So that's the background for this paper. They want to look at the dynamics of water attaching to these spores and see if they could learn something about it, the hygroscopic property, which has not been studied before. So they use a technology called environmental scanning electron microscopy.
You're going to tell us who the authors are, aren't you? You haven't done it yet.
Yeah, I did.
You did? Sorry.
I did. Hassett, Mary Beth Hassett.
Yeah, you did.
Mark Fisher, and Nicholas Money. No problem.
My brain isn't working
I'm sure the authors love to be mentioned twice. It's no problem. All right. Environmental scanning electron microscopy is used. And what this does, it allows you to look at untreated biological samples. So they're not treated with anything. So you preserve the surface chemistry of the spores. And then you can look at the spores in real time and see what's going on. They can visualize the water formation in real time because the scanning.
The big deal here, which runs counter anything I ever heard about electron microscopes, is that you can look at the specimen while it's wet.
Right.
So you make a special chamber in the microscope and you control the moisture.
Right.
And that I thought was impossible, but apparently it can be done.
Yeah, so you can watch.
It's revolutionary.
You can watch moisture condensing on this, right? So environmental scanning electron microscopy. So they collect mushrooms. That's the first step. So they actually pick some mushrooms. They do this in Ohio, and they collect 7 different species of mushrooms. They have a lovely photo of each species in the paper, and these include mushrooms whose spores are discharged in the way that we've just described by the Buller drop and the adaxial drop, and they also include a puffball and an earth star, and in these, the spores are expelled from the fruiting body by the impact of raindrops. Everyone's probably seen a puffball. You squeeze it and poof, out come the spores, a puff of spores. Well, it turns out that when rain hits that's a signal for the spores to leave. Makes sense, right? And they expel it.
Essentially, the puffball becomes a bellows.
Right.
It's like a bellows. It's just the right consistency. And the covering is tough. So it stays around for quite a while, and it takes quite a bit of rain to displace all the spores that are in it. But eventually, that's what happens.
Right. So they have different kinds of spores. They have spores that are spherical. They have spores with a spiny surface and a smooth surface. They have elliptical spores. So all different kinds. And then they put these mushrooms above a piece of aluminum, which is going to be used as the surface for the electron microscopy. They let the mushrooms stay there for a couple of hours, and the spores fall onto the surface. For the puffballs, they squeeze them. They say, we gently squeeze them to cause puffing. I thought that was funny, and they release the spores. And then they put these, they're called aluminum stubs on which the spores are. They put it in a cooling chamber where the temperature is kept constant, but they can alter the vapor pressure, which will then change the relative humidity. So constant temperature, altered vapor pressure, and therefore they can regulate the humidity from below 100% to just above 100%. So what do they find? So these spores that are ejected from the mushroom gill, these are called ballistosporic. I like that, having ballisto in there. For the ballistosporic basidiospores, they could see water droplets forming on the hilar appendix and on the adaxial surface between relative humidities of 101 and 102%. And they have lovely images showing a dry spore, the initial droplet formation, and then the droplets get bigger and bigger and bigger. And they can reduce the relative humidity below 100%, and then the water evaporates. So they can actually do an experiment where they raise and lower the relative humidity of the same spores and do images, and they have a nice graph of this. They can make a droplet, they will dehydrate it and rehydrate, and they can go over and over again on the same spore. Now, these droplets, at the Buller's droplet and the adaxial droplet, You never see them forming separately. They are always either one or the other. And the adaxial droplets tend to be much larger, and they often exceeded the diameter of the spore. And these droplets can enclose multiple spores as well. So
by the way, I've been fussing around how to say adaxial, and it occurred to me we should say what it means. It's a botanical term. It means it's situated on the side toward the axis or the stem. That's in a leaf, of course. So this is situated towards the hilar appendix.
Right. Yeah, you'll have to look at these pictures. It'll become perfectly clear. And this is open access, so you can look at Figure 1. But I'll probably include a picture in the image of the podcast so that you can look at it without going to the journal. So they can see by this electron microscope the relative humidities of over 100%. They could see water droplets condensing on the spores, encasing the spore in multiple spores. They can lower the relative humidity and the water evaporates, and they can do it over and over.
By the way, what is it? What is more than 100% humidity?
101, 102%.
Yeah, what's that?
Well, if you increase the vapor pressure, you can achieve that, right?
I see. I guess so. Yeah, of course.
Which you wouldn't have, although in a moment we're going to see that this can happen in clouds, but normally here on the surface of the Earth, this doesn't happen, yeah. But under their experimental conditions, they did it. But below 100, the water didn't condense on the spores, so it's really interesting. Now, in the spores that are not shot out of the mushroom, the non-ballistosporic basidiospores, at relative humidities over 100%, these spores did accumulate water, but it was very different. It formed a thin shell around the spore, not discrete droplets. They call this kind of a halo. All right, so the two kinds of spores, the ones that are shot out of the mushroom as opposed to the ones that go out when, like in a puffball, when raindrops hit it, they react differently with the water. So the authors think that what they're seeing in the electron microscope is what they call drop expansion, is basically what is happening when Buller's drop is created on a spore in a mushroom. But theirs is quite different. And what they're thinking is that they're using spores that are already discharged, right? So the ones that drop onto their electron micrograph surface have been discharged. And when the spore is discharged, the two droplets, the Buller's droplet and the adaxial droplet, they coalesce. And this, they think, spreads mannitol sugar over a large portion of the spore surface. And then when water condenses in their EM studies, it forms as a single drop, not two drops, because you've got sugar all over the surface. So the distribution of sugar in the original spore is important for getting these two droplets, whereas in their experiment, you could call it a used spore. It's got mannitol all over the surface, so it just forms a single large drop, and it gets bigger and bigger because the spores are not moving and it's not evaporating. So they say that in clouds, you can get supersaturation such that the relative humidity exceeds 100%. And they think that such conditions would, in a cloud, if there were spores in the cloud, and there certainly are, that would allow the formation of droplets on suspended spores. So in their experiment, again, you need to have relative humidities over 100%, and apparently this can happen in a cloud. So what does this all have to do with rainfall? Well, rainfall starts when water condenses on the surface of aerosolized particles, and these typically are greater than 2 microns in diameter.
Ice nuclei.
Ice nuclei, they're over 2 microns in diameter, and these then coalesce with smaller droplets. They merge with each other, and this merging eventually produces a water droplet. All right, so it's not condensation of water vapor, but it's actually droplets attaching to these particles and the particles merging and forming larger and larger particles until a rain droplet is formed. And they think these hygroscopic basidiospores are just the right particles for forming these large droplets with condensed water vapor on them. And they think they're effective nuclei of raindrops. And the droplets they see in their electron microscope are consistent with them being part of the formation process of a raindrop. So their hypothesis is quite interesting. So basically you have in the mushroom, the spores develop. They're ejected by this water-driven process. The water evaporates, and then some of the spores may make their way into clouds where they now can serve as nuclei for condensation of water at the right relative humidity. The water droplet will now encompass the entire spore and multiple spores. It's now different from what it was in the actual mushroom, and this can stimulate precipitation, basically. So
I imagine that this is just a physical attachment of the moisture to the surface of the spore,
right?
Because there's no mannitol being made by the spore.
But there is
right.
But there is mannitol on the surface, right? And
I see.
It was distributed originally when the spore was ejected and the water flowed over the surface of the spore. Their hypothesis is the mannitol was distributed over the entire surface, and that's what allows it to attract large drops, and make an actual water saturated particle.
That's clever.
And so, how would that benefit the fungus?
Yes, so that's a good point. They make
I can tell you.
Well, they say
you don't get mushrooms unless it rains.
Right, but so they say it's clearly a positive feedback mechanism, right? You get fungal growth stimulated by rain, that disperses spores, you enhance precipitation, but they say there is no adaptive significance to the putative effect of spores on cloud formation. They think it's a consequence of the dispersal mechanism that happens to have benefits beyond dispersing spores. So they don't think there's any connection, although it helps the fungi. And then, of course, if you don't have enough rain, if let's say there's climate change, you have reduced rainfall in tropical ecosystems, you get an inhibition of fungal growth, you get inhibition of spore release, and then the drought would even worsen because you need the spores to make rainfall, right? So I don't know if they're right about that there's no adaptive significance. I don't know how they can conclude that, but the negative effects in drought are quite clear because the rain is stimulated by fungal spores, and if you don't have enough rain to make enough spores, it gets even worse. So
right.
I think that's very cool. It's just amazing.
It is cool, but what's next? I mean, this is not based on the rain formation is based more on probability, a likely conjecture, but there is no experiment behind it.
No,
right?
Of course not. I mean, these are.
What are we going to do? Have spread spores over the cloud and see if it rains?
I guess you could do that, right?
I don't know if the EPA would love that.
Yeah, right. Yes. Yeah.
But I guess you could do it in a laboratory in a contained environment where can't you create clouds in a certain box and see what happens?
Yeah, I suppose you could do that.
Because I must say that there's a bit of a jump in this paper. I mean,
of course, absolutely,
very likely, very plausible, but if you want to nail it down, you got to go a little bit beyond it, I think.
But their contribution is to show that it is possible for these fungal spores to continue to form these droplets once they're released from the mushroom. So it's plausible.
No, it's very plausible.
Yeah.
Yeah. This episode of TWiM is also brought to you by ASM Biodefense and Emerging Diseases Research Meeting. Early bird registration rates are going to end on January 14th. So you need to decide whether you're going to this meeting or not and go ahead and register. This meeting is taking place in February 8th through the 10th. It's going to be in Arlington, Virginia. And this meeting is the premier event to discuss how to defend against bioterrorism and how to shape the future biodefense research agenda. There's also quite a few sessions on emerging diseases as well. I will be there doing a TWiM. Not sure which day yet, but I'll be there. Come by and check out the session. If you want to register for the meeting and learn more about what's going on in Arlington from February 8th through the 10th, you can go to asm.org/biodefense2016. So biodefense 2016, all one word. asm.org, Biodefense 2016.
So can I tell you a little about Mary Beth, the first author? Mary Beth Hassett. She received her bachelor's degree from Ohio University in environmental and plant biology, and there she started working on fungi. She was in a plant cell wall lab and studied fungal enzymes that had a potential application in biofuels. And then she decided, after taking a mycology class, she decided to go to Miami University and study for a master's degree with Nicholas Money. And there, after she got her master's degree, she, of course, was hooked on the science and wanted to stay for her PhD and start working on this project. And she describes that one of the most exciting moments in her career to date was sitting at the microscope, this scanning electron microscope, and watching these Spore-drops forming on the basidiospores. She also wants to give credit to their long-term collaborator, Mark Fisher. He's at the University of Mount St. Joseph in Cincinnati and has been a long-time collaborator, and he's especially good at making these beautiful scientific figures that really illustrate the principles that we're talking about here. So he's a middle author on the paper. And then she also wants to point out she's not the only fungal enthusiast in her family. Her grandfather first introduced her to edible fungi, and he was an avid mushroom hunter, like you, Elio. And she writes that one of her favorite possessions is her grandfather's field guide, which has notes about the different mushrooms that he discovered out on his explorations.
That's very cool.
Yeah.
What's she going to do next?
She didn't say. We weren't able to talk because of the holiday, so I wasn't able to ask her more questions.
I'm curious about the next step in this story, you know.
Right. Yeah.
If they're going to try and make clouds in the laboratory and test it, that's cool.
Or find a collaborator
right.
Who works in that realm.
Wow, a field guide. That's cool that she found that.
I wonder which one it is. I used to collect such field guides.
Yeah.
Mm-hmm.
Elio, have you collected mushrooms lately? Probably not, right?
Well, it hasn't rained in San Diego, so it hasn't rained because there are no mushrooms, and there are no mushrooms because it hasn't rained.
Catch-22. It's going to get worse.
Let me say something about Nick Money, the senior investigator. He is I've met him. He's a very interesting guy. He's a fantastic writer. He's written several books on mushrooms and fungi. The last one is called Mushroom, Oxford University Press. It's a great book. I really recommend it. And he also has a book called The Mysterious World of Mushrooms, Molds, and Mycologists, plus some other books, Carpet Monsters and Killer Spores. Cool. So his writing is just fabulous. So I really recommend this book.
Yeah. I'm going to look it up.
You yourself have written a book on mushrooms, yes?
I have written a book on mushrooms. It's called In the Company of Mushrooms. And it does deal a little bit, in general terms, it deals with these issues as well.
Mm-hmm. In the Company of Mushrooms. Wonderful. All right. We have a few emails for you. They're very interesting. First one is from Chris, who writes, I have a fun question for your panel about Mars. I was watching NASA talk about how Mars's atmosphere was stripped away and was wondering about Mars bugs. Here are some fun, probable facts I learned. When life was starting on Earth 3.8 billion years ago, Mars had an atmosphere about the same size as Earth and had liquid water in lakes and streams all over the surface. So Earth and Mars look a lot alike. At 3.5 billion years ago, Mars's dynamo cooled down and its magnetic field stopped. Then the high solar activity of the sun sent strong solar winds that stripped away Mars's atmosphere, and Mars turned into the icy desert we see today. The one place NASA would look for microbial life today is in Mars's aquifers. If they found some, they would like to know if the genetics are essentially different between Mars and Earth. So let's assume that there was some cross-pollination between planets when both were watery, by asteroids throwing Mars and Earth rocks back and forth. My question is, would it be possible for Mars microbes and viruses to infect Mars colonists, or would the genetics be too diverged after 3 or so billion years? Will Martians have to put up don't drink the water signs? It's a good question.
It's a very good question. First of all, there's no evidence yet that there is life on Mars extant. There's very little evidence that there has ever been, but much less about life today. But let's play the game. Let's assume that somebody finds it. My dime would go on what he just said, namely, what Chris pointed out, the genomes are going to be very different. After all, they would be selected for very different skills. If there are no Martians around, why would a microbe learn how to infect a Martian, leave alone an Earthling? What do you think?
So the assumption would be that there are microbes still growing and have grown for many, many years on Mars. In the absence of life that we know here on Earth, I would agree that it's probably too diverged to infect any of us. What do you think, Michele?
A key is each microbe needs to be able to make 2 microbes, and to do that, they need to be able to get the nutrients to build a new cell. And so if their nutrient pool is very different from ours, that might be a roadblock.
Yeah. I mean, the assumption here, which he says, is that Earth and Mars, at one point were very similar. And then they diverged after Mars lost its atmosphere. So
didn't they just make a movie called The Martian? I haven't seen it. Have you seen that movie?
I read the book.
But an astronaut being stranded on Mars. Yeah. Maybe we should ask him. What did he see?
Well, he grew some food, but he did it indoors in his artificial environment, But they don't talk about any of that because we don't know. I don't know. My guess would be that it's not likely that you would get infected there, but as Michele says, if the requirements for growth are similar enough, it's possible that, a microbe could accidentally infect and just by accident could infect, right? I mean, that's the premise of the Andromeda strain, right? A strain came from another planet, but it just happened to be able to get what it needed from humans, and so it infected us. So I don't know. My vote is no, but who knows? It's really an interesting question. And probably there are other people around who have thought about it more than we have, right?
If it did happen, it would probably be pretty nasty. I'm thinking, for example, the rabies virus is adapted to live in bats and doesn't cause terrible disease. But if it accidentally infects us, we get overwhelming infection. So
any of these zoonotic infections we get by accident where the microbe is not adapted, it causes nasty infections for sure. So it could be, yes, I agree. If it did happen, it would probably be pretty nasty,
In that cases, think about tetanus, disease by a bug that lives in soil, minds its own business.
Exactly.
It makes a toxin that affects mammals.
Right.
That makes very little sense.
Yeah, you could argue that that's been around in an environment that has had, not humans, but other animals.
Nematodes.
Hey.
Pack hunters. That could be. So, Elio, would that classify as a Talmudic question? Because we don't have an answer.
Absolutely. This is very Talmudic.
Talmudic. All right.
The question is more interesting than the answer.
Yeah, it is. All right. Andrea writes, hello again. Here's another one of those articles I find a little strange, but what do I know? And she sends a link to an article from Campus News at the University of Nottingham, where they talk about a university study where they have found antimicrobial properties in the extracts of cockroaches and locusts, which they say could lead to future antimicrobials. And Andrea continues, I guess it kind of makes sense, but I would never have thought of this. I bet you already know about this, don't you?
Oh, boy.
The rains have returned to Seattle. Yippee. Well, you can, people look for antimicrobials in almost anything. I'm not surprised. Whether or not it's of any use is another story, right?
I think we had a podcast about a beetle that was being used to harvest a particular compound.
Right. The next one's from Arthur, who writes, hello, TWiMers. I have been an avid listener on and off for many years since my undergraduate days. I started...
What does it mean, off? What's this off?
Yeah, really. Thank you. I started off learning about viruses from you and now love to listen for interesting tidbits on other microbes. I work on Mycoplasma pneumoniae, and my graduate school has one of the largest mycoplasma research groups in the U.S. I think you've discussed the synthetic genome cell before, a mycoplasma species, but I don't think that I've seen a full session about the smallest known self-replicating prokaryotes. I'm a bit biased, but I know some wonderful researchers who would love to do a podcast if there would be interest. Keep on being awesome, and thanks for making my bench time entertaining. So who has one of the largest mycoplasma research groups in the U.S.? Does anyone know? Where would that be?
I don't know off the top of my head.
Yeah, so we could do an episode on some mycoplasma. Why don't you send us, tell us where to look, Art? That'd be fine. Yeah, we haven't really done Other than the synthetic genome, we have not.
They have a mode of locomotion that's wild.
Yeah.
They send out sort of like a grappling hook and then reel it in.
Mm-hmm. Oh, that's cool.
Something like, well, not quite. That's more like pili motility.
Right.
Anyhow, it's fascinating.
I like their antigenic variation of the human adapted mycoplasma.
Okay.
I remember they have a tiny organ of locomotion, which when detached from the cell, still moves.
Wow.
Cool.
I think that's right.
Well,
I think that's right. I may be off some, but...
The next one is from Sean, who writes, "Hi, everyone. I've been listening to TWiM for the last few months during my more monotonous lab tasks and have learned so much more about microbiology in systems I'm not familiar with. I would have never thought to read some of the papers shared on the show. However, as a plant pathologist, I keep waiting for the day you put us on the map and bring one of us on. I've only been listening since number 87, so maybe I missed it. If not, I know Michele lives about an hour southeast from a university with a pretty decent plant pathology department. What would that be?
That would be Michigan State.
Oh, yeah?
Home of the Spartans.
Plant pathology.
Yeah.
I agree with him. It's fascinating. I mean, the plants have immune systems that don't resemble, some resemble actually what happens in animals.
Right.
So it's really fascinating. Some don't.
Okay, so we'll put that on our list too, plant pathology. And I know someone at Michigan State. Vic, right?
Yeah, Victorita. And our microbe textbook co-author, Hema.
She's at Michigan State. She's not a plant pathologist, though, right?
No, she's not.
Okay. And she doesn't work on mycoplasma, does she?
No.
Okay.
Well, she works on electricity, conduction in bacteria, nanowires and things. We should have her on.
Yeah, definitely. For sure. Okay, Anthony, we have two more. Anthony writes, though exposure to Aspergillus by immunocompetent people generally is without drama, and that's something we mentioned on a recent episode. The air is full of spores. We inhale them all the time. That's not true for penguins. Air quality is a big question in maintaining these birds in captivity. If climate change brings an increase in Aspergillus spores to the Arctic, this won't be good for penguins. And Anthony sends a link to an article. This is very cool. It's a PDF, which we will post. It's published by the Association of Zoos and Aquariums, and it's called The Penguin Care Manual. How to take care of penguins. It's a multi-page document all about it, and apparently you have to make sure that the air is good. You have to filter the air, and in particular, they don't like to have Aspergillus fumigatus spores. The penguins don't like it. So who would have known, right? Thank you, Anthony. All right, here's one more. And Elio, this is quite interesting, and you will have to explain this at the end, okay? It's from Jeffrey, who writes, Drs. Schaechter, Racaniello, Swanson, Schmidt, and any guests that I might have missed. In honor of a section that shows up on your show on occasion, I thought that I might send you an actual Talmudic question that I posed and partially answered for another podcast a year or so ago. And here's the question. Why were the Hebrews unable to take their leavening with them when they left Egypt under Moses' leadership? During the 7 days of Passover, the commemoration of the Exodus, Jews are forbidden to eat chametz. Is that how you say it, Elio? Chametz?
Yep, that's correct.
Although often translated as leavened bread, the word seems to derive from a root for sour, indicating that it was a sourdough-type food. The word is probably better translated as foods or drinks containing leaven. Exodus 12:15, which includes beer. The production of beer and bread were intermingled in ancient Egypt and probably most of the ancient Middle East. Why bring this up on TWiM? Because chametz is the result of seor or microbial fermentation. What could be more appropriate than an actual Talmudic question about microbiology? Elio, seor, is that how you say it? Seor?
Which? I'm sorry, say again?
S-E'-O-R. How would you say that?
Seor.
Seor, okay. Now, Exodus 12:15 provided the injunction against leaven. Quote, for 7 days you shall eat matzah. Matzah is unleavened bread, flour, and water mixed and baked within 20 minutes, not enough time for yeast production to kick in, but not the reason. A reason was given in Exodus 13:8. quote, "And you shall explain to your son on that day, it is because of what the Lord did for me when I went free from Egypt." End of quote. But honestly, that isn't much of a reason. Part of another reason was given in Exodus 12:34, where it stated that, quote, "They took their dough before it was leavened," unquote. And in 12:39, where it stated that, quote, "They baked matzah of the dough, which they brought forth out of Egypt, for it was not leavened, since they were thrust out of Egypt and could not delay," end quote. That on its surface might seem reasonable enough, but any of us who have baked bread and or know anything about bread microbiology know that there is something wrong about this whole situation. If they took their dough with them, then it already contained this seor, starter. The bread might have collapsed on them if they waited too long to cook it, but they didn't leave this seor behind. That could only have happened had they only brought their grain or flour with them and mixed it with water while they were setting up camp, and that is not what the text says. And there's another complication. Exodus 12:39 implied that they couldn't bring their leavening with them because they were thrust out of Egypt and could not delay. In other words, they didn't have enough time. Yet the Hebrews had sufficient time to gather jewelry from the Egyptians, enough supplies to build the tabernacle and an altar, and their livestock. They even had time to bring their netting boards, Michele, bound in the clothes on their shoulders. We know that the Egyptians, and Hebrews living in Egypt, used two main types of starter, a baked yeast cake for storage and basically a sourdough starter for continuous use. As most people who have dealt with sourdough starters know, these starters, with their often unique tastes, were cherished family possessions being maintained like a family pet and passed down from generation to generation. They were also mobile. Many American prospectors in Alaska in the late 19th, early 20th century kept them as a cherished part of their minimal possessions in their camps. That means that if the Hebrews had time to gather other possessions, then they had time to pack their seor and bring it along. In fact, if they brought their dough along, then they were technically bringing their seor. So that brings us back to the question of why leavened bread was forbidden during Passover. I propose that the reason was cultural rather than physical, i.e. a matter of time. The Hebrews that left Egypt were depicted as belonging to many tribes. Their journey through the wilderness was a depiction of their unification into one overall tribe, the Jews. Seorim, sourdough starters, were cultural heritage because they were handed down from generation to generation. They represented the past. Discarding this seorim and eating matzah for 7 days until they could make more through wild capture was an act of discarding the past as various Hebraic tribes and starting a new life as one larger Jewish tribe. I suspect that the Rosh Hashanah ceremony of Tashlik, when one tosses breadcrumbs into a body of water to cast away the sins of the previous year, is a similar act. Here, too, the seor and shametz serve as symbols of one's past, but delving into the connection further is, I fear, a Talmudic question for another time. This might seem a strange excursion for the TWiM podcast, but when I started to piece it together, I found it to be another fascinating example of how microbiology can serve as a scaffold for understanding cultures long since vanished from the earth. Thanks for the podcast, Jeffrey.
Wow.
You got that, Elio?
This is fascinating. I never contemplated that question. I always assumed that it was Israelites did not eat bread because they didn't have time to make it. But I think that Jeffrey destroys that notion.
Yeah.
I think he says, they had time. They had time on their hands. It doesn't take that long for bread to rise. So I think we're left with a mystery here. And Jeffrey, I would really encourage you to submit a piece for our blog, Small Things Considered. I'd be very happy to contemplate publishing such a thing.
Yeah, basically, he's got the framework for it right here.
Yeah. But what you said, just put down what you already said.
Yep. And if I apologize if I mispronounced any of these words, and I'm happy to hear a correction. Thank you all for your letters. And you can find this episode of TWiM and all the others at iTunes and at microbeworld.org/TWiM. And we love hearing from you. You can write to twim@microbe.tv. We have a new email there, twim@microbe.tv. I want to thank everyone for participating, not only here today, but for the past 2015. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure. What fun.
I also want to thank Michael Schmidt, who isn't here today, but has been with us for much of the year. He's at the Medical University of South Carolina. And I'm Vincent Racaniello. You can find me at virology.ws. That is the crew of TWiM. I want to thank them all for their dedication to promulgating information about microbial sciences. I also want to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.